Hello everyone, and welcome to the second in our series of anniversary lectures to celebrate the 75 years of Great Ormond Street Institute of Child Health. I'm Claire Smith. I'm going to be hosting this event today with my colleague Daniel Brayson, and we will be hosting the question and answer session at the end of these two lectures that we've got today. So if you have any questions for either of our speakers, you can submit them on our Slido app. So go to slido.com and enter the code ICH75present. And now I'm going to hand over to our Institute Director, Professor Rosalind Smith, for introducing the speakers for today. Thank you.
So a very warm welcome, colleagues and friends, to the second in our series of 75th anniversary lectures. We got off to a great start yesterday with Tim Cole on his 75th birthday, presenting his historical perspective of the Institute of Child Health and also his really very important work with the child growth charts, which millions and millions of children and families around the world have benefited from over the past 2 or 3 decades. So as an illustration of the kind of breadth of what we do at the Institute of Child Health, we have 2 very different speakers today representing the present. And I'm very delighted that David Long and Manju Kurian can present their work. So as Claire has said, please put your questions as you think of them into Slido. We'll take questions for both speakers at the end of Manju's talk. But as soon as you have a question for David, pop it in the Slido and we'll make sure we can get that to asked. So go to Slido and it's ICH75 present. And you can also endorse other people's questions. So I'd also highlight that clearly we're using our live YouTube video to stream this, and those talks will all be recorded and available to look and view at your leisure later on. So David Long is Professor of Paediatric Nephrology and a Wellcome Trust Investigator based in Developmental Biology and Cancer at the Institute of Child Health. And his interest is in renal and vascular biology. His initial research experience was at ICH as an MRC-funded PhD student before he did some postdoctoral work in the US at the Baylor College of Medicine and at the University of Florida. And he returned to ICH to establish his own research group, supported by a senior fellowship from Kidney Research UK and an MRC New Investigator Award. And his group is a strong kidney translational research mission, aiming to understand mechanisms which underlie kidney disease in children and subsequently in adults, with the goal of translating those into patient benefit. So we're very excited to have you as a speaker, David, and looking forward to your lecture on innovative technologies to identify new treatment targets for kidney disease.
Okay. Can you see my screen, yeah?
Can you make it full screen?
Yeah. Okay. Thank you very much for the introduction, Ros. It's really a great pleasure to give one of the talks here for the 75th anniversary lecture at Great Ormond Street Institute of Child Health. And I'm going to tell you about some of our recent work, which is using innovative technologies to identify new treatment targets for kidney disease. So I've been asked to tell you a little bit about my previous experience at ICH. So to do this, I'm just going to take you back to when I had joined the Institute in 1999. So it seems a very long time ago now. This was the the technology available in mobile phones, and we were very worried about the millennium bug and every computer going wrong in the year 2000. But this was the first time that I actually entered the steps at Great Ormond Street Institute of Child Health here. And I was a very naive 21-year-old who was looking to do a PhD and start to do some research. And I was particularly interested in coming to the Institute of Child Health because I was keen on the translation that we have between basic science and groundbreaking research that goes on in the lab and taking this into the clinical setting. And really, Great Ormond Street and the Institute is an ideal place for us to be doing this. And it's really been the ethos of the Institute while I've been here for the whole 20 years. So I answered this advert in 1999 to do an MRC studentship, and I was awarded the studentship and given the choice to either work on neural development, heart development, genetics, or kidney disease. And I decided to work on kidney disease, and I worked together with my PhD supervisor, Adrian Wolf, who's still my mentor 20 years later, and Haitao Yun, who was a PhD scientist who taught me a lot of techniques that I learned in the lab. So why was I interested in the kidney? Well, there were two main reasons. Firstly, there's a very scientific interest, and the kidney is a very interesting organ to study, but also it has a huge clinical need in terms of patient care and therapy. So the kidney does a very simple job. It filters the blood and excretes any waste product as urine, but actually it's a very complicated organ to do this, what seems a very simple task. And to achieve this, we have in each human kidney over 1 million structures called nephrons, which is this structure here. And this consists of a ball of cells called a glomerulus, which is the filtration apparatus of the kidney, and this tubular network, which goes ahead and helps to reabsorb any nutrients that we may still want in the body, while the rest is excreted as urine. And so this was a very interesting system for me to study, and also it's a very important clinical need. We have over 10% of people worldwide that have some degree of kidney disease, and this is caused by various different causes, such as genetic disorders, such as cystic kidneys, damage to the glomerulus, that filtration apparatus, and also systemic insults such as diabetes. And there are different degrees of kidney injury that you can see here. So here in this initial 3 stages of kidney disease, your filtration starts to gradually decline, but this can be controlled by pharmaceutical drugs such as antihypertensives, and also changing our lifestyles. However, when we get to stage 5, really this is a problem for any patients, and their renal function has really declined, and they require either dialysis or transplantation. And we have over 2.5 million people worldwide that require dialysis and transplantation, and a similar number who actually can't access this treatment at the moment. And there's 70,000 people in the UK, and over 1,000 of these are children. So there's a real need for new treatments for kidney disease. We don't have enough transplants to go around, and dialysis is a very an intervention which is very difficult for patients and also very costly, and also is associated with other things such as an increase of cardiovascular disease. So I decided that I wanted to work on the kidney, and so the next 15 years after I'd done my PhD was really spent developing my research team at Great Ormond Street. And I went across to America, as Roz said, for 2 years to work with a clinician scientist called Rick Johnson, who was interested in metabolic syndrome and diabetes. And then this slide really represents about 15 years of work, really roller coaster of getting the group together and applying for lots of grants. And then, fortunately, in the end, I got fellowships from Kidney Research UK, MRC and Wellcome, which have allowed me to develop my own group. And this is what the group looks like today. And we have a mission to understand and develop new treatments for both children with kidney disease and the adults that they become. So in my talk, I'm going to tell you about 3 very quick stories of how we're using new technologies to identify new treatments for kidney disease. I'm going to tell you about new ways that we're imaging the kidney, which have allowed us to identify lymphatics as a new target for cystic kidneys. I'm going to tell you about how we're using genetic barcoding of individual cells to hunt for new treatment targets for childhood glomerular disease. And finally, I'm going to tell you how we're using human three-dimensional cell models in a dish to really understand how diabetic kidney disease works. So I'm going to tell you first about imaging and how that's been helping us to understand cystic kidneys. So when I started in 1999, it's like the main technique that we used to image the kidney was histology. And really this means taking the kidney, cutting into small sections, and then having a look under a light microscope to what the kidney looks like. And this is still critical for clinical diagnosis. The problem with histology and for us understanding processes that are going on in the whole kidney is that it's two-dimensional and it really just gives a snapshot of the whole organ. So really over the last few years, led by a PhD student in my lab, Daniel Jaffrey, we've really been developing techniques to look at three-dimensional imaging of whole mouse kidneys. And we've also now been applying this to large sections of human kidneys. And this is a technique that Daniel has developed. So really we isolated the kidney, we pre-treat it with various chemicals and immunolabel it for the cells or the molecule that we're interested in. We then make the kidney transparent and clear the kidney using various solvents. And then we look at it under a confocal or a two-photon microscopy. And this allows us to actually look at the whole kidney in 3D. So we've used this technique to visualize a specific cell in the kidney, which are called lymphatics. And to tell you a little bit about lymphatics, here they are in green in the kidney here. And really they're the waste disposal system of the kidney. And that they act to remove excessive fluid and immune cells. They're very poorly characterized, both in health and disease kidneys. And they're an ideal system really to study by this technique of three-dimensional imaging. The lymphatics are very rare in the kidney. So if you do histology, you sometimes find sections where you can't find any lymphatics. And so really to get an idea of how the system, the whole lymphatics are working, this 3D imaging is ideal. And they're also a potential treatment target for kidney disease, in particular cystic kidneys. So in cystic kidneys, what happens is here on the left is a normal kidney, but a mutation in a particular gene makes the kidney become cystic and these fluid-filled cysts. And this is one of the major causes of kidney disease, both in children and in adults. And what happens in cystic kidney disease is that we have these process of excess fluid and also an accumulation of immune cells. So this is why we think the lymphatics may be very important in this disease. So this video gives you an idea of the imaging that we can do and how we can visualize the lymphatics. So this is a whole mouse kidney, and you can see that it's labeled for two molecules called PROX1, which is here in purple, and also Li1, which you'll see here in green, which label the lymphatics. And you can see where these two colours overlap, which is here in yellow, is where our lymphatic system is. And this technique can be used, and we can then computer analyse these branching networks that are occurring of the lymphatics. So we did this in polycystic kidney disease, and we used a mouse model of polycystic kidney disease. Here on the top here, you can see this is a normal mouse. In here is these balls of these glomerular filtration apparatus I told you about. And here is the tubules of the kidney. And this is a very early stage mouse with polycystic kidney disease. You can see it has dilated tubules, but the structure is relatively intact. So we're looking at a very early stage of disease here. And we did our imaging. Here's what you can see. This is a normal kidney, and this is the cystic one. You can see that this lymphatic network is very stunted. And we were able to quantify these parameters looking at the vessel diameter, the volume of lymphatics, and in these two graphs, and the number of branches that we had. And you can see that the lymphatics were impaired in these early stages of cystic kidneys. And so this analysis allowed us to go on to trying to start to develop lymphatic therapy for cystic kidney disease. And so for this, we've been given a molecule called VEGF-C, which enhances lymphatic migration, stabilization of lymphatics and survival. And we gave this to our mice that have polycystic kidney disease. Here you can see this is normal. This is our cystic mouse. And here on the left, on the right, sorry, is the mouse that's been treated with VEGF-C. And you can see there's a significant reduction in the size of the kidney. And this was also seen on histology. So this is the cysts that we can see in a normal cystic mouse. And here is when we give them the treatment with VEGF-C. And we were able to measure the kidney body weight ratio, which is the clinical parameter that people use for polycystic kidney disease, and also look at the renal function, which is measured by blood urea nitrogen, and they were both reduced with this therapy. So I've told you a little bit of how we're using imaging to identify lymphatics as a target for cystic kidney disease. So I'm now going to tell you about how we're using the genetic barcoding of individual cells to hunt for treatment targets for childhood glomerular disease. And I'm just going to take you back to this picture that I showed you earlier. And the work that I told you about in VEGF-C is really basic science research, which we're hoping to translate to the clinic. And the story that I'm going to tell you now is going in the other direction. So we've got hints from the clinic and my clinical colleagues that I work with at GOSICH, which has then been able to take back to the lab and hopefully back it later into the patient for therapy. So this is some work that's been done with two of my colleagues, Aoife Walters, who's a clinician at GOSICH, and also Jenny Chandler, who's just been awarded her own Kidney Research UK Fellowship and is a postdoc in my lab. And before I tell you a bit about all the work that's done, I need to tell you a bit more about some details of the kidney. So in my earlier slide, I told you about this ball of cells, which is the filtration apparatus of the kidney. But actually, those ball of cells is much more complicated. So this is a close-up of what that filtration apparatus look like. It has a blood vessel, an endothelium, which sits next to a basement membrane, and also these wavy cells, which are epithelial cells called podocytes, which I'm always told look like octopuses. I'm not sure whether that's the case really, but they have a slit diaphragm here between each podocyte, and this allows the molecules from the blood out into the urine. And in kidney disease, what happens is that these slits become bigger and more molecules are allowed out. So what Aoife and Jenny have been doing is they've been taking patient samples to identify genes which may be involved in childhood glomerular disease. And what they found is that the majority are found in the podocyte cells. And in particular, there's a gene called Wilms tumor, or WT1, which is mutated in about 6% of cases of childhood glomerular disease. And it's a hotspot at a particular amino acid at 394, where in a lot of patients, an arginine is substituted for a tryptophan. And what we've been able to do is actually use this fact to generate and use a mouse model of this mutation and identify new treatments potentially for disease. So this is what happens when you get childhood glomerular disease. Your podocyte cells become damaged and filtration becomes leaky, leading to albuminuria in the urine. Your glomeruli becomes sclerotic, and this eventually causes renal failure. And this is what happens in the mouse model. So at 4 weeks of age, you can see that we start to get albuminuria, and this is continued to 6 and 8 weeks. But at this 4-week time point, the glomerulus is relatively normal, and it only becomes destroyed at 8 weeks of after disease progression. So this gives us a therapeutic window here to potentially treat these mice, but also find early drivers of disease that may hold therapeutic potential. So this is what we've been doing. And what Jenny has done is she's isolated glomeruli from 4-week-old early disease kidney tissue. And she does this by putting magnetic beads through the mice, which accumulate in the glomeruli of the kidney. She then uses a magnet to isolate the glomeruli and put these glomeruli into a suspension. She can then digest these down further into single cells. So these are the podocytes, the endothelial cells, which we find in the glomerulus. And we're able to use a technique called single cell sequencing, which allows us to take each individual cell and read the genetic expression one by one. And more clever people than me are able to analyze this data and use data analysis and bioinformatics to cluster the cell types together and analyze them. And this is some of the data that Jenny's got here. You can see this is the data from the glomeruli of the mice she's been looking at. You can see here in red are all the podocytes that have been able to be isolated, but we also have endothelial cells, smooth muscle cells, and other cells, mesangial cells, which I've also found in the glomerulus. And Jenny can take the podocytes that she's looked at and look for differential expression analysis between the wild type and the mutant podocytes. And this has allowed Jenny to identify various different molecules which we're now looking into in the lab. Some of them are involved in maintaining the shape of the podocyte, which is important for the glomerular filtration barrier. Others are involved in glomerular cell communication, and others are associated with the risk of kidney disease. And in Jenny's current work, she's going to actually be looking at using and therapeutically targeting these molecules to see if she can improve the progression of renal disease. Okay, so my final part of the talk is really looking at human three-dimensional cell models in the dish to understand diabetic kidney disease. So we're changing tacks a little bit. The first two examples were examples of genetic kidney disease. Here I'm looking at a more systemic insult, which causes kidney disease, but primarily actually in adults. So diabetes is a real problem in the UK. We have about 3.8 million people in the UK that have diabetes. It costs about 10% of the NHS budget. And the majority of these costs are really due to complications that are associated with diabetes. And this includes kidney disease. So about 20% of all people that have kidney disease is due to diabetes. Also changes in the heart, in eyes, and also in nerves. And this is caused by changes that occur in blood vessels due to high sugar levels and high glucose. And so really we desperately need treatment strategies that can potentially protect blood vessels in diabetes. And this is one of the areas that our lab is also working on. So one of the problems is that we don't really have any good human models to really understand diabetic vascular complications. But a student in my lab, Gideon Pomerantz, working with a group in Vienna led by Joseph Penninger, has established this model of using stem cells to actually look at diabetic vascular complications in the lab. So here you take a stem cell, which is a cell which can then be differentiated to many different cell types in the body. And you give it various chemicals, which here we're giving chemicals which want to make it into a vascular cell. And then we aggregate this cell into a gel. And this is done around 6 days after the protocol started. The cells then start to sprout like blood vessels. And by day 18, you're able to actually get what's called a three-dimensional organoid in a dish, which represents what blood vessels look like. And you can see that they have a normal structure of blood vessels. So they have an endothelium, they have a pericyte, which is a supporting cell on the blood vessel, and also a basement membrane. And we're able to image these. What Gideon's done is that he's taken a dye called Evans blue, and he's put this into the organoid to see where it goes. And you can see that the blood vessels here are labeled with a molecule called CD31. And the dye is in pink. So you can see here all the blood vessels have a lumen, and the dye that Gideon's injected appears actually in those lumens of the blood vessels. So what we've been doing with Joseph Penninger's lab is we've been exposing our vascular organoids to diabetic conditions. And you can see this is what happens to the organoid in diabetes here. The basement membrane becomes thicker, exactly what happens to vessels in patients with diabetes. And we've been able to do that genetic barcoding I told you about to identify what molecules are changed. And excitingly, one of the molecules that I worked on in my PhD, a molecule called angiopoietin-2, was one of the big hits that we got during this experiment. And just to tell you about that molecule, it's really a molecule which works together with another molecule called angiopoietin-1, and they work as a pair. In health, you have lots of angiopoietin-1, which is a good guy, and this leads to vessels being nice and stable. But in diabetes, we have more of the angiopoietin-2, which is a baddie, and this leads to vessels becoming destabilized. And in mice, we've been able to give angiopoietin-1 therapy. So this is a normal mouse, this is a mouse with diabetes, and this is a mouse with diabetes and angiopoietin-1 therapy. And you can see that the levels of albuminuria, a measure of vascular leakage, improve when we give angiopoietin-1 therapy. And so we're hoping now to actually look at manipulating angiopoietins in this diabetic human model that we've got. So to conclude, I've told you about innovations in imaging, genetics and human cellular models, which are able to provide avenues to identify new treatment targets for kidney disease, both in children and adults. We're really in a very exciting discovery phase for kidney research at the moment, and we're identifying a lot of new treatment targets which will be helpful for patients in the future. And really the challenge for the next 5 to 10 years is to really translate these basic science discoveries into actual treatments which will benefit the patients that we see at Great Ormond Street and all around the world that have kidney disease. So this is just to acknowledge the people that have helped with the work. It's really a very collaborative approach to research that we have in our lab. These are the many people that are in my group, and particularly shout out to Paul Wynyard, Maria and Karen Price, who have been with me for the journey really from the beginning. Also our collaborators at Great Ormond Street, ICH, UCL and externally, and also our many funders. Thank you very much for listening, and I'm happy to take any questions.
So, David, that was terrific. You and Tim have set the bar very high for the quality of talks for celebrating our 75th anniversary. So if I could just say to the audience, pop your questions for David into the Slido and we will get to them. But that was a beautiful, clear presentation. So I now want to turn to Manju Kurian, who I met first before I came to ICH, and it's been a great pleasure to get to know Manju better in ICH. So she is Professor of Neurogenetics and NIHR Research Professor at this institute, and also a consultant paediatric neurologist at Great Ormond Street Hospital. She graduated from Cambridge University and trained in paediatrics before specialising in paediatric neurology. And she undertook a PhD in Birmingham, investigating the molecular genetic basis of childhood neurological disorders, and then moved to UCL after this time to establish herself as an independent principal investigator, firstly through a Wellcome Trust intermediate clinical fellowship, and now the NIHR research professorship, which she was awarded in 2015. And in the same year, she had a very good 2017, Manju. She was awarded the Jules Thorne Award for Biomedical Research, so huge achievements. So we're looking forward to your talk also, Manju, which is entitled Translational Research at ICH, From Bench to Bedside for Neurodegenerative Disorders. Manju.
Thank you very much, Ros, for a lovely introduction. Can you see my screen?
Yes, just put it on. Yeah, perfect.
Great. So thank you very much, Ros, for such a lovely introduction. And just like David, it's an absolute privilege to be given the chance to speak to you all today about my research. So as Ros said, I'm going to talk for the next 20 minutes about translational research at GOSICH, really from the bench to bedside for a number of childhood brain conditions. So just to give you a background of what I'm going to talk about for the next 15 to 20 minutes, I'll give you a brief background of my clinical and research career. I'll focus on my main research goals. I'll talk to you about how the UCL Great Ormond Street Institute of Child Health has really facilitated my research. And I'm going to just tell you some success stories and how we've managed to bring or are bringing new treatments to our very precious patients. And I'm going to also talk about some exciting future perspectives and novel therapies. So just as a background, I'm of Indian origin. I was born in a very small town in Kerala in the south of India to Keralan parents who immigrated to the UK. We eventually ended up in Newcastle, where I went to school. And after school, I then went to Cambridge to study medicine. And after medicine, I started my clinical training and really loved paediatrics and worked in a number of different paediatric hospitals, including Guy's and St Thomas's and the Children's Hospital in Dublin, where I got a really good training in paediatrics, and then really decided to then focus in on the brain, which had always fascinated me since I was a child, to subspecialize as a paediatric neurologist. And at the end of my training, I ended up in Birmingham, where I completed my training. And you can see there's a picture of me doing clinic with two very esteemed colleagues, Dr. Hardev Pal and Dr. Martin Smith. We used to run a movement disorder clinic there. And it was really here that I forged my career interest, because I really realized that it was very frustrating being in clinic, because the vast majority of children that we saw in that time neither had a diagnosis, nor did they have any satisfactory treatments for their neurological disorder and movement disorder. And it felt very frustrating, both for the families and as a clinician, to be part of that. So as I said, this clinic really formulated my main aims. And my main aim is a very patient-centric bedside to bench approach, really, that I really am back again from the bench back to the bedside, really focused on the child and the family, and really focused again on what the children and families are asking us in clinic. And the main things they ask us are things like, why has this happened to my child? Families want answers to why things have happened. How has this happened to my child? You know, what's the underlying cause and mechanism of what's happened to my child? And what can you do as my doctor to try and cure or improve my child's life? So really, in a nutshell, from the clinic that I did in Birmingham, these research aims really formed for me. And essentially what I'm trying to do is develop better, or what we call precision therapies, precise therapies for rare neurological disorders. And when I tell people this, many people say, why are you working on rare diseases? You're only going to benefit a small proportion of the population. And why are you working on neurological disorders? They're often lacking hope. There's really nothing for them. And that's exactly why I want to do this. So firstly, I would argue that rare diseases are very important, and they're not so rare. So all the statistics show that even though individually they may be rare, there are at least 6,000 rare diseases. 1 in 17 people will suffer from a rare disease at some point in their lives. And importantly, many rare diseases, almost 4/5 of them, are genetic in origin. For me, as a pediatrician, it's important to know that 75% of rare diseases actually affect children. And very sadly, only 1/3 of children with rare disease survive after their 5th birthday. So you can see these important reasons why it's important to work on rare disease. And GOSICH is the perfect centre to be able to do this. And through this, we're able to drive precision medicines. And many of the precision medicines that we're developing and the approaches that we're taking have such relevance to adult onset disorders, such as Parkinson's and Alzheimer's disease. And I'll try and show you how in a few examples in my talk. So what do I mean by precision medicine? Well, precision medicine, as I've shown here, has been in many of the major media outlets. You know, it's a real kind of hot topic at the minute. And it's defined as medical care that's designed to optimize efficiency or therapeutic benefit for a particular group of patients based on their underlying genetic or molecular profiling. So what their genes and proteins tell us about them and how we can group patients. And really the approach is to try and use this information about a person's profile to try and curate their medical treatment by combining really good research with detailed information about their individual's kind of makeup to try and improve a patient's life. And really that concept of precision medicine is really at the heart of everything we do at the GOSICH. So I would argue as a paediatric neurologist that the concept of precision medicine has been around for a very, very long time. I'm just going to show you a video of a patient where she has a condition that's been known for decades, where we've been able to instigate a precision therapy. So this young lady was thought to have a form of cerebral palsy, and you can see why that she's walking very high on her tiptoes, and it can look like cerebral palsy. But the clue to her really was that her condition varied throughout the day and was progressive over time, which really isn't something that you see particularly in cerebral palsy. we were able to work out that she had an underlying genetic problem that affect a very key pathway that produces important neurochemicals that control movement, known as dopamine, and also your fright and flight response, norepinephrine and epinephrine. So she has a block, as I've shown here, in the pathway, which means she can't produce enough dopamine. But you can see even from this pathway that if you're able to bypass the block and treat with L-DOPA, you might be able to have some good results. And just like that, this young lady was given a trial of L-DOPA, levodopa that we use for Parkinson's disease. And within a very short period of time, she became normal with her walking. She was able to walk without any tiptoe walking. And then she started to ride a bike. And you'll see in a video very shortly how excellent her walking and stable her running is. And you can see here her running very nicely, completely different, and even riding a bike from a child that could barely walk. So just by understanding the underlying mechanism of disease, we were able to understand what treatment would work for her. And this is an absolutely brilliant example of something that's been known for decades, known as Sagawa's disease, where we have a clear understanding of the medical problem. We're able to give something orally. The L-DOPA or levodopa can be given by mouth. It crosses into the brain and it reaches the right brain cells that produce dopamine. So we're able to replenish the dopamine in this child and completely rescue all her symptoms and restore her back to normality. So you can see how this story particularly inspires me to think, well, if we can do this for Sagawa's disease and this particular condition, there's no reason we can't do it for every single childhood neurological movement disorder. So with this in mind, this was really my driving move to come to GOSICH. I did a PhD in Birmingham over about 3 years where I found new genes, and then I moved to GOSICH, and as Ros mentioned, was able to secure a number of fellowships from the Wellcome Trust, NIHR, and the Sir Jules Thorne Charitable Trust. And this allowed me to build my research group. You can see the members of my research group here, as well as a number of other grants from the MRC and various charities, including Great Ormond Street Hospital Children's Charity, to really push my research into the direction of finding answers for patients and developing new treatments. So here we are again, back to my original research aim, which is really trying very hard to try and understand patient disease and developing new treatments. And by coming to this institute, it really has allowed me to develop that. I'm able to find new genes, which allows us to find answers for families. We use state-of-the-art laboratory models that I'll show you to understand disease, and we're developing new pioneering treatments for our families. So just to kind of recap what I mean by genetics and gene answers, we all have cells in the same way as bricks make up a house, our cells make up our human body. And as I can show you here, within the center of a cell is the core nucleus. And the nucleus is our core hub of our genetic material, where our genetic material is coiled into these special structures called chromosomes. And within that are sections of DNA that code for single genes that code for a characteristic in our human body. And over the last 10 years since I've been in research, we've been very lucky to be part of either the actual discoverers of a number of genetic conditions or helping to understand the genetic condition and what sort of conditions they cause in our patients. And I've made a list of over 50 diseases that we've managed to either find or delineate and understand. But really, we've moved beyond that gene discovery now, and it's not enough there. What we really want to do is find better treatments for our patients. And as I've shown here, there's a number of different approaches that we're trying to embed for our patients with movement disorders. And this may involve some dietary manipulation. It might be that we're trying to reduce toxic molecules. It may be that we're trying to electrically stimulate the brain. We may be developing new gene therapies. We may be developing new drug therapies. And with the genetic therapies, I'm talking particularly about things like gene therapy. I'm talking about antisense therapies, and you may have heard of CRISPR correction, where we correct the faulty mutation. And all of these depend on the underlying cause. And I'm going to show you a few examples of this where we've managed to produce some really life-changing results for the patients that we look after. So particularly, I'm going to tell you about 3 different disorders, as I've highlighted, and how a precision or personalized treatment approach has led to some significant benefit for our patients. So first example I'm going to just focus on is within this kind of treatment approach, something called deep brain stimulation, which is an approach by which you can electrically stimulate the brain with continuous electric stimulation to allow you to try and modify your motor or movement output. So during my time here at GOSICH ICH, we discovered a group of patients who were diagnosed, many of them as having cerebral palsy because they had an abnormal pattern of walking, as shown in this young man, but actually they had a genetic disease that we were able to find and delineate. You can see this young man was very debilitated, and then he had deep brain stimulation, and you can see the very, very striking improvement. His walking pattern has near normalized, and he's done really very well. So we managed to change his diagnosis. We gave his family a definitive answer for his underlying condition, which was a genetic answer, which has huge implications for his wider family, for his parents, and also has allowed us to give us a very, very effective treatment, which has led to a marked improvement in his walking and his motor symptoms. But not every disease we've found has been as, dare I say, as easy or straightforward as that. And for many diseases, we lack understanding of the disease. And the brain is such a hard organ to target to try and get the cells that we need in order to understand treatments. We can't easily access brain cells. And even if we could, it's really hard to try and grow those brain cells in a culture system within our laboratories to try and help us understand disease. So instead, we use this very clever technique called stem cell biology. And what this means is that we take samples from our patients, usually skin cells that the patients provide for other diagnostic tests, and we use those skin cells and we're able to convert them into stem cells, which are the type of cells in your body that can form any type of cells. And using certain important and special factors, we're able to then convert them into brain cells. And we can then use these brain cells by looking under a microscope or doing special tests in the laboratory to try and understand the underlying disease mechanisms. And we can use that then to find new drugs, test gene therapies, find new markers of disease, and things like cell replacement therapy, where we replace faulty cells with healthy cells. And to give you an example of where we've done this, I want to show you one of the first genes that I found. And this is a lovely young lady who has a genetic fault in this gene that causes this form of infantile Parkinson's. And you'll see from her video that she clearly has features very similar to adult Parkinson's, where she has this real slowness of movement. She's very bright and she knows what she wants to do, that her movements are very slow, and she finds it very hard to express her facial expression. And you can see here that her neuroimaging really shows features very similar and actually even more severe than adult Parkinson's disease. So we didn't really understand this disease very well, and in order to understand it better, we used her skin cells and converted them into brain cells, and not just brain cells, but also into organoid models, which are like small brains in a dish. So you can see here with the red and green that they really do look like brain cells that have been grown from her skin cells. They have these long projections, which are very classical of brain cells or neurons. And now we can make them into a three-dimensional structure, which really look like a part of a brain that we're able to study. But most importantly, we can use these models to develop new treatments. So we've used this particular model to test with gene therapy. And we had a striking result in that we were able to completely restore everything that was wrong in the cell as a result of the genetic fault, which means that really gene therapy is a very, very promising treatment for children with this form of disease. And we're very now quickly moving to clinical trial, hopefully in the very near future, for children like this patient and many others that have this infantile forms of Parkinsonism. And what we plan to do is to inject the gene directly into this part of her brain called the midbrain, which is where the center of movement is controlled. And our hope is that by replacing the faulty gene with a healthy working copy of the gene, we can improve her movement and give her a better quality of life and make her less prone to all the complications of such a disease. So really that's our hope. But you may say to me then, is this really a reality for children with movement disorders? So I'm going to tell you the story of one of our patients where it's an absolute reality. So I want you to meet this lovely little lady who I've known since she was an infant. And she has a condition called aromatic L-amino acid decarboxylase deficiency. And all you really need to know about that is that it's a condition that really reduces your levels of dopamine and serotonin, which are very important chemicals that are involved in controlling movement and mood. And as a result, she has these very painful crises where her eyes will deviate upwards like this, and she's very still, she can't move. She was very globally delayed and very, very floppy. She has now had gene therapy where the correct gene has been put back in her brain. You can see this is the actual surgery where the gene therapy is injected, and it's hit the right spot in the midbrain, exactly where we want to hit it. And I see her very regularly in clinic now, and you can see her here. This is her here, just a few months after treatment, and she's gone from a child that couldn't hold her head to now being able to sit. And you can see here, then a few months later, her sitting is even better, and she's developed some words. And at last review, just a few weeks ago, she's able to start taking steps. So she's gone from a child that could not lift her head to a child that's now taking steps. So she's made significant developmental gains. She's got words, she can speak. Her eating and drinking is now safe. She doesn't need a tube in her tummy to help her feed. And because of the success of the gene therapy that's been put in her brain, we've been able to take away nearly all her medications that she was previously on that were needed. So she's done miraculously well, really. And it really shows us, I think, how gene therapy is a real possibility for children with these complex and previously untreatable movement disorders. So how has GOSICH helped my research? Well, I hope it's obvious, but just to spell it out, having Great Ormond Street Hospital right next door to our research base is fantastic. So the hospital and the research is synergistic to each other. I'm surrounded by lots of like-minded clinician scientists, people like David Long and many others, and also basic translational researchers who really drive this goal of bringing novel therapies to our patients. I've been really well mentored by many people here to allow my goals to develop, and also now involved in, as a more senior academic, in mentoring. We attract some of the best trainees, and to be able to train the next generation is a real privilege. And there's a huge encouragement towards outward facingness. We have strong partnerships within our institute, but also more widely across UCL, and also both nationally and internationally. Also, just to mention, we're now based in the Zaid Centre for Research into Rare Diseases in Children, which is just fantastic. We have our clinic rooms on one side and we have our research rooms on the other. You can see our brilliant laboratory space. This is Katie, our RA, who's showing our lovely research space so nicely. We have beautiful laboratory space where all my research team, as shown here, are able to undertake their experiments, and fantastic tissue culture space where we grow our stem cells. So I'll finish there really by saying you can show all this media coverage that we've had really shows how translational and important rare diseases, how it touches everyone in society, and how some of the breakthroughs that we have far-reaching effects, not just for rare neurological disorders, but much wider within society. It's a really exciting time to be a scientist and neurologist. I think the landscape for childhood movement disorders is really changing. They've gone from untreatable, undiagnosable diseases to a forum where diagnosis is likely, understanding disease is likely. I hope I've shown you that there's lots of novel treatments on the horizon, and really our ultimate goal, and it will be for the rest of my career here, will be to reduce disability, reduce the risk of death, and improve quality of life for all our precious patients. Thank you very much, and I'm very happy to take questions.
So Manju, as always, that was a tour de force. So thank you so much. It was really inspirational, and your videos just tell the whole story. So I'm going to hand over to Claire and Dan to lead the questions. We've already got a number in on the Slido.
Yeah, amazing talk, both Daniel, David and Manju. Really enjoyed it. So yeah, if anyone has any questions, you can still submit them on the Slido app at #ICH75 present, if you want to go there. Also, just to everybody watching, to really help us with our future public engagement activities, please subscribe. There's a little button just down here. If you subscribe to our channel, then that will really help us to be able to support more of these types of events. So I'm going to go straight to Daniel, because he's out and about in the Institute, and he's found someone who's got a question for you. I think he might be in the ZCR actually, so we might be able to see some of that. Daniel, are you there?
Hi everyone, I've come to the ZCR, as you can see, and I have someone who would very much like to ask a question. This is Alice. Alice, don't be in the light because we won't be able to see your face. So if you'd like to fire away.
Yeah, hiya. First of all, thank you for the very fascinating talks, both of you. I just wanted to ask, why did you choose a career in health sciences? Like, what made you do it? Was it something at school that struck with you, something that happened as a kid? I was just interested to see and hear why this career. Thank you.
Manju, should we go to you first?
Well, was the question Korean Alzheimer's? Yeah. Why did it? I mean, so. That's a good question. I think I've always been fascinated by the brain since I was, a child, really. I think it has such, the brain is the central control. I know that Daniel will argue with me that the kidney is the most kind of important organ, but I would argue that the brain is the most important organ, and it's so complex, just from, tiny movements to kind of quite complex thoughts. I think it's really fascinating. After my training, I really wanted to be a paediatrician. I really love looking after children, and within that, all my favourite attachments were looking after patients with neurological brain disorders. The neurodegeneration is particularly appealing to me because I think it's so hard. So it's always nice to have a challenge, and I really hope by the end of my career that I've changed the landscape from being one where people see these children as being undiagnosable, untreatable, to diagnosable and treatable. So it's the challenge of that really has inspired me, I think.
Great. And David, do you have an answer to that question as well?
Yes, so I guess why I became interested in science generally was because of a teacher really at school really inspired me, and it was about when I was about 14, really, when I started to become interested in science. And really it was a teacher at school who was, their main job was actually to teach us rugby, but in their spare time, they also taught us human biology. And really, he really inspired me in terms of actually understanding what is going on in our actual bodies and what happens in disease. And really, that was a very simple message that he gave me at the age of 14, which really stuck with me. And so that's why I became interested in science and doing the research that I do.
Great, thank you both. So really great answers. So Dan, I know you're going to go and try and find someone else to ask questions, so I'll get back to the slide for now. I have a really nice comment actually on there about hearing today about the work that my colleagues are doing here at ICH. Makes me enormously proud to work here, which I think is a really nice comment. And I've got two technical questions that have come in, so I'm going to go to David first. Very interesting talk, David, thanks. I was wondering whether you considered spatial transcriptomics in combination with the confocal imaging.
Yes, we have. So that's something that we're working on at the moment. So yeah, in our next sort of papers and our next phase of work, it's something that we're going to be working on.
Okay, thanks. And one for you, Manju. Long-term L-DOPA treatment is known to cause side effects such as dyskinesia. How is this mitigated when treating children, if at all?
That's a really good question. So in actual fact, if you, some of the conditions, the dopaminergic synthesis conditions, many of them you don't see dyskinesia. So the condition I showed with the Sagawas, dyskinesias are very, very rare. Some of the other conditions you do see dyskinesias, and so we use other drugs, things like amantadine can really help control those L-DOPA related dyskinesias. Sometimes you have to switch to a different dopaminergic medication, such as the dopaminergic agonists, things like bromocriptine and pramipexole and reticotine patches we use. For those children with neurodegenerative disorders, I very much see the dyskinesias as a huge part of the condition that initially they'll have an L-DOPA response, but over time that response wanes, or you see on-off effects, and that then requires either switching to a different type of agent or using another drug like amantadine to help control those dyskinesias and help the L-DOPA tolerability.
Great, thank you. And now I've got a question for both of you, but we'll go to David first. How do you think the COVID pandemic has affected and will continue to affect research at ICH over the next couple of years?
So yeah, like with everything, the COVID pandemic has affected research. I think it's taught us that we can do flexible working. I think that's one important thing, that some of the jobs that we all were doing at the office or lab can be done at home. So I think that's been an improvement. It's also, I think in our lab, it's really helped that people have really had to organise their experiments. So they haven't really been able to come in all the time. We've had to work in shifts and in less days a week. And so we've really had to focus our experiments, which I think has been an important thing. The thing that I've missed during COVID is really the interaction that we haven't been able to have, particularly face-to-face. And this is really important particularly for more junior researchers. It's like really when I was starting my career, most of the best ideas I had were when I was having a cup of coffee with someone, and we've missed that. And I think that's important that we try and get that back. And so our younger researchers are able to have those interactions going forward.
And Manju, how about you? I mean, particularly on the point that David just raised about interactions with people, have you had any thoughts about how you can integrate that into your group, like moving forward? Or
yeah, I mean, I think that's a huge issue. I mean, so when COVID hit for us, because we have long-term cultures, some of our stem cell cultures, when we're growing the neurons, that the protocols are 70 plus days, so we lost a lot of material at the start of COVID. And, obviously the lab work, we've had to adapt very quickly. So the minute we were allowed back, shift work, shift patterns, as David says, being very, very organized. Moving, the remote working has huge advantages in that, there's a huge efficiency to Zoom and Teams meetings, but at the same time, the big lack is that face-to-face contact. So now that restrictions have lifted a bit, we have face-to-face, with safety measures in place, we have face-to-face lab meetings, and it is hugely different and hugely beneficial. We get lots of interaction between group members, which is really important, and that fuels everyone's research forward within the group. So having that is really important. I gave my first talk at the CRIC not that long ago, face-to-face, with all measures in place, and I remembered how fantastic it was to be at a physical conference. I hope that things continue to improve, because I think whatever of being a professor, I think as a junior researcher, to be able to have that face-to-face lecture experience, networking is really, really important. So they're the things that I think we need to work on as we move forward.
That's great, thanks. So someone who is face-to-face, although masked up, is Daniel. He's found someone else for a question, so I'll hand over to him.
Hi, everybody. Now, carrying on our tour of ICH, we're in a lab, and I have a very hard-working researcher here, Sam, and he's going to ask his question.
Hi, great talks by both speakers. My question is that, other than the pandemic, what is the biggest challenge your research is facing right now?
Manju, do you want to go first? I know you see.
That's a really good question. So I would say funding for rare diseases was a challenge, but I think that the tables have turned slightly on that. As Ros mentioned, I've been very lucky to get quite major mainstream funding for my research. And I think that's partly because of the understanding that how much rare diseases can contribute to biology, understanding pathophysiology of not just those disorders, but of more common disorders, as I kind of use the example of rare childhood neurodegenerative disorders, helping us understand Parkinson's and Alzheimer's. So that's a challenge. I think keeping everything really fresh and moving forward with science is really important. So that's something that I constantly find a challenge. So making sure I'm well read on up-to-date papers, and that my lab is moving forward and not stuck with techniques that are 10 years old, that we're right on point when it comes to kind of things that are new and exciting. I think that's a big challenge, but it's a challenge I enjoy, so it's okay.
Great. And David, do you have an answer to that question? Your biggest challenge right now?
Yeah, well, I agree with Manju that funding has always been a key challenge for everyone going forward. In the kidney area, it's been a disease that perhaps hasn't had as much funding as others, and that's always been a challenge for us looking forward. And I think now with the increased amount of people that do have kidney disease, hopefully that will be something that will go forward, and we'll be able to have more funding to look at that disorders. I agree with Manju, trying to keep up to date with the literature is tricky now. It's there's so many things going on. There's so much big data that people have now produced and to analyze. And I think that's something that there's also new skills for the scientists, really, not just lab work, but also having computational skills ready to look at all of that data. So I think that's something going forward.
Fantastic, thank you. I think I've got a few more questions, but I think we're going to have to wrap it up there. So I'll hand back to Ros just to finish up. And also don't forget, subscribe to the YouTube channel, please. Thank you.
So thanks very much to all of you for your participation and questions. We had 200 people yesterday, and I'm sure we've got a similar number today. Of course, thank you to our two speakers who have really fantastically presented some of the variety of the important work that's undertaken here at the Institute of Child Health. So next week, on the 29th, on Monday at 3 o'clock, we have The Future, which is Julian Baratu and Alice Guistacchini, who are going to also do a sort of double act and present two talks during the hour. So many thanks again to our speakers and to you, and see you next week.